Uh, we're all familiar with bleeding. This can happen after a car accident and there's a lot of blood being lost. This can also happen on the battlefield in regions of conflict. In fact, bleeding is the leading cause of potentially preventable deaths in young people worldwide, making it a major problem. Currently, the gold standard therapy to treat major bleeding once the patient reaches the hospital or medical tent or trauma resuscitation area is the transfusion of platelets. Now, platelets are anucleate cells that circulate in the body where they detect and adhere to sites of vascular damage. This is where they begin to activate and generate the clot. So in situations of hemorrhage, platelets are transfused to stop bleeding. There are nearly 2 million platelets transfused annually in the US alone, making platelet transfusion one of the most common and widely accessible cell therapies. Not only are platelets transfused in situations of active bleeding, but platelets can also be transfused as a replacement strategy if a patient has low platelet numbers or dysfunctional platelets. Moving back to the context of active bleeding, one of the major problems is that platelet transfusions are really not always effective. So in some cases of major bleeding, despite the transfusion of platelets into the bleeding patient, there can be continued uncontrolled bleeding, representing significant mortality. And so one of the major questions of our lab is, can we engineer enhanced donor platelets to address issues that are encountered during major bleeding and really enhance the functionality of donor platelets? Now, one of the most common approaches available to engineer desirable cell attributes is a process called transfection, which is fundamental to biotechnology. So the question we pose is, can we directly transfect platelets? Now, when we look to the literature, there has been no reported successful transfection of platelets with corresponding protein expression, representing a major gap in blood biology and medicine. And attempts have been made using chemical methods, including cationic polymers, and physical methods such as electroporation, none with success in expressing protein in these platelets. In fact, in our own internal studies where we look at the common commercial reagents, ribojuice and lipofectamine, and we deliver an mRNA encoding a luminescent reporter enzyme into platelets, we get no observable protein expression. This corroborates findings in the literature. And so this begs another core question to our lab. Is there a transfection approach amenable for platelet engineering? And being a nanomedicine community, we're all familiar with lipid nanoparticle technology, which is being used in leading mRNA vaccines for COVID-19. And so a core hypothesis to this project is, can we leverage lipid nanoparticle technology to efficiently deliver mRNA into donor platelets to achieve uptake of the genetic material, resulting in the expression of exogenous proteins that can modify the platelets desirably? Ultimately, the goal is to engineer platelets with these lipid nanoparticles to express procoagulant proteins that might allow us to better control bleeding once the platelets are transfused. And so the next thing is, can we actually transfect platelets using LNPs? And so over the past 2 years, we spent a large amount of time developing a lipid nanoparticle transfection platform for platelets. And so going back to a previous figure, what we've been able to do is take lipid nanoparticles, expose them to platelets, and really for the first time achieve robust expression of a luminescent reporter enzyme directly in platelets, a major milestone for the project. What we have done over the past few months is rapidly try to identify an optimized LNP formulation that can enhance the expression of exogenous proteins in platelets above our pre-optimized LNP. And what we've been able to do is achieve that optimized lipid nanoparticle formulation, resulting in almost a 9-fold increase in optimization or in expression levels. So the two stories that I'm excited to share with you today is first, how did we identify this platelet optimized formulation? The second is, we know that lipid nanoparticles can have some toxicity to the cells. So if we are exposing platelets which are highly sensitive to their environment, if we are exposing these platelets to lipid nanoparticles, do these platelets still retain their function, which would be critical for use as a therapeutic? And so going to the first story, I want to provide a brief overview of our methodology. The first thing we do is we receive pooled donor platelets from the Canadian Blood Services Blood for Research facility here in Vancouver. We then move these platelets into a transfection buffer and incubate them with mRNA LNPs. The general components of a lipid nanoparticle include a PEG lipid, cholesterol, and notably an ionizable and helper lipid. We then assess modification after 4 hours. And so how did we actually optimize these lipid nanoparticles? Well, we all know that the helper lipid and ionizable lipid components of a lipid nanoparticle can have a dramatic effect on the cell type specificity, on the toxicity, and the degree of protein expression. So can we identify an optimal ionizable lipid and helper lipid combination that is ideal for platelet transfection? And so to do this, we screened a library of ionizable lipids and treated our platelets. And what we found is that we could achieve robust protein expression in our platelets above the dotted line, which represents the standard pre-optimized LNP formulation. We then did the same process for the helper lipid component. Again, the dotted line representing the pre-optimized formulation. Collectively, not too much of a dramatic increase in expression. However, when we systematically exchange the ionizable lipid identified in the previous screen and the helper lipid identified in the helper lipid screen, when we do a combinatorial test, we can identify a LNP formulation that gives us almost a 5-fold increase over the pre-optimized formulation, leading us to conclude that we have successfully identified a platelet optimized LNP formulation for ex vivo transfection. We then can use this optimized lipid nanoparticle to deliver mRNA encoding another protein, so a different protein altogether. In this case, a firefly luciferase reporter protein. And we can see this trend is still present between our pre-optimized and our optimized LNP, although not to the same degree. So this is wonderful. We can now successfully engineer platelets to express proteins of interest. However, are lipid nanoparticles detrimental to platelet function? Do these LNP engineered platelets still retain their function? And so to address that question, we can do 2 tests. The first time that I will talk about involves identifying if LNP engineered platelets still respond to their physiological agonists. So in a bleeding situation, when platelets begin to clot, there are many mediators in the forming clot that can drive platelet activation, and the platelets then can contribute to clot strength. So the question is, do these LNP engineered platelets, would they respond to agonists? And we can test this by setting baselines. And so in this situation, we're using flow cytometry and looking at a marker of platelet activation, CD62P. And the first 2 bars indicate our untreated platelets with and without activation with thrombin. And as we can see, there's a dramatic increase, as expected, in platelet activation after we treat with thrombin. So do we observe this with our LNP treated platelets? And in fact, this same trend is also present in our LNP treated platelets, suggesting that our LNP treated platelets are still responsive to thrombin. They're still responsive to their physiological agonists. The next test we can do is to see if these platelets would still be functional by modeling an actual transfusion event, asking the question, do engineered platelets still clot? Now to do this, we use an ex vivo or in vitro tool to assess clot formation kinetics, and we can simulate a transfusion. And what we do in this situation is we take normal patient blood, which coagulates normally and we can use as a benchmark. We then simulate a patient that has suffered major bleeding, and then we can test the coagulation ability of these 2 different bloods. We find that, as expected, our normal patient blood or whole blood, we get large amounts of clot firmness. We get a very strong clot. However, with the patient that has suffered major bleeding, we get impaired clot formation. We get reduced clot firmness, demonstrated by the difference between the red and the gray lines on this graph. We can then simulate the in vitro transfusion of LNP engineered platelets or untreated control platelets into the patient with major bleeding. And when we supplement these untreated and LNP treated platelets, we can specifically isolate the platelet contribution to clot firmness. Looking at these parameters more closely in this model, we can look at the clot formation time, so how long it takes to generate a clot, and the maximum clot firmness, being the strength of the clot. And in both cases, between the untreated and LNP-treated platelets, we do not observe a statistical difference, although there is a trend that there could be some slight impairment, which is a consideration for future studies. So before I conclude, I'd like to thank all members of the Kastrup lab, the Devine lab, and our collaborators, Dr. Peter Kohls and Eric Jan. I'd also like to thank all of our funding sources, as well as NMIN for this opportunity. So to conclude, we have shown really for the first time that platelets can be directly engineered. We have identified an optimized mRNA LNP formulation that specifically enables robust protein expression in platelets. We've also demonstrated using common clinical tools that these LNP engineered platelets still retain their function. So what's our next step? Well, we want to express hemostatic proteins or therapeutic proteins in these platelets to cause a functional change to the platelets. Can we express a procoagulant protein in platelets that then enable them to have stronger coagulation abilities once they're transfused into a bleeding patient? Beyond this, we think that these engineered platelets really can become a platform technology, introducing a variety of new platelet products. We hope that these new platelets can lead to improved transfusions, and we know platelets can be important for circulating throughout the body, making opportunities for targeted therapeutics and drug delivery. We also hope that these LNP engineered platelets can be used as a research tool to address fundamental basic platelet biology questions. Thank you, everyone.
I could jump in the conversation and say that, again, it was a great presentation. What will be the amount of LNPs that were transfected in your platelets, for instance?
Yeah, that's a great question. And so I think by that you're referring to the amount of uptake. And one thing that we know is that if we, in addition to looking at the levels of protein expression in our studies, we also look at a couple other parameters. One of the most notable being the degree of uptake. So how many LNPs are getting into these platelets? And we can do this by using a fluorescently labeled mRNA. And so the more fluorescence you get from your platelet, the more LNP uptake. And in this figure that I'm presenting, we can see the commercial reagents achieve high levels of mRNA uptake into the platelets. However, there's no corresponding protein expression. But when we look at the lipid nanoparticles, for example, we can see that there's actually lower levels of uptake, but it's that fine-tuning balance between the effect that we have on our platelets and the ability to deliver an adequate amount of nucleic acid. And so it's these lipid nanoparticles that are, you could say, safe enough to deliver mRNA into platelets without damaging them, and enabling this expression.
Okay. And for instance, do you think that this will impact the transfection will impact on the lifetime or the shelf lifetime of the platelets? I'm not sure what is actually the lifetime that you can store them, but do you think there will be any impact of that?
Yeah, so that's also a phenomenal question. It's something that we're definitely thinking about. We know that currently platelet units that would be used for transfusion into a bleeding patient, for example, they're stored for 5 to 7 days. In the case of engineered platelets, we do have some planned stability studies. So that will be storing the engineered platelets and then looking at their coagulability over time and comparing them to standard controlled platelets under the same storage conditions. But one thing that is really promising about this data is that we are seeing acute levels of expression. So within a really short time frame of 4 hours, we get dramatic levels of protein expression. So although we may not be engineering platelets at the beginning of their, before they are stored, we can at any point in the storage lifetime take these platelets, engineer them right before they would be transfused or used in any meaningful way, and we can engineer the expression of protein within 4 hours. This obviously depends on will have to be evaluated in future studies when we look at additional proteins, or we'll have to look at the individual expression kinetics of the potentially therapeutic proteins to make sure that they would be useful in the respective situation.
Great, thanks for your answer. That was nice. Marshall, do we have any other questions?
Yeah, we actually have a written question from Sirazam Manira. who asks if you're at liberty to explain the methodology of nanoparticle preparation.
Yeah, it's pretty well reported in the literature what how many lipid nanoparticles are made, and in this case, I can provide, I guess, a brief summary, which is that what we do is we prepare our different mixtures. So this would be an aqueous mixture that includes our mRNA, and then we prepare a lipid mixture, and that includes the 4 different components: an ionizable lipid, a helper lipid, a pegylated lipid, and then cholesterol. And then using the T-junction methodology, which is very well published on, we then mix these 2 components, and then through the neutralization of the pH, we get the generation of lipid nanoparticles. And although it's not included in any of my supplementary material, we can and we have confirmed that our LNPs fall within the expected range for size, for encapsulation efficiencies of the nucleic acids. And so I hope that answers your question.